My name is Aviv. I'm a postdoc at Richard Bonneau's group in NYU. And today I will talk to you about some of my recent research about the regulatory network controlling Th17 specification that we recently published in Cell. And so Th17 cells normally function in immunity to protect against bacteria and fungi at mucosal barriers. But when they are dysregulated, they are also implicated with multiple autoimmune diseases, including psoriasis, multiple sclerosis, and Crohn's disease, very common autoimmune diseases. So going into this project a few years ago, we had some prior knowledge to begin with. We knew some of the environmental factors that play a role in differentiating this subset of T helper cells. For example, some of the cytokines involved. We knew some of the transcription factors that were core to this differentiation process, including the lineage specifying factor, rho gamma T here in the middle. We also knew some of the effector genes that are important for the lineage and how they are regulated by these 5 core TFs here. But of course, this is just a small part of a very large regulatory network, and a lot remains to be discovered, both for transcription factors, effector genes, and environmental factors that control this lineage. And so we decided to take an integrative genomics approach to examine the Th17 specification. We used mice as a model system. We purified naive CD4 cells. We then in vitro differentiated them into T helper 17 cells, which is the cells we want to study. We then performed ChIP-seq and knockout RNA-seq on select transcription factors that are important for the lineage. We also complemented that with transcriptomic data, particularly a time series following the differentiation of these cells from naive CD4 cells, as well as a public microarray data, the InGen data that has microarrays for many of the immune cell types in the immune system of mouse. And at the end, what we wanted to do is to use these orthogonal sources of information that all can give you some indication of a regulatory interaction between transcription factors and genes, and we want them to put them together into a regulatory network that is very context-specific into relating the regulatory events controlling this differentiation process, and is highly accurate due to the combination of orthogonal data sources. This was a very tight collaboration. 3 labs were involved. The Bonneau lab, who I'm a part of, who is a computational lab, the Littman lab, that is an immunology lab, and the Myers lab at Hudson Alpha, Alabama, who do a lot of sequencing and are really experts in everything next-generation sequence related. And I want to acknowledge Maria Ciofani, a very talented postdoc from the Littman lab, who was the other side of this project, and we worked in close collaboration for quite a few years to get it together. And so all of the results I'll show you today are part of what we published recently. And so before I jump into the network, I actually want us to focus on the ChIP-seq data, because I think there is a lot of mechanistic understanding that you can gain just by looking at the ChIP-seq data. So what you have here is one genomic region that is important for Th17 cells. These are the IL-17A and IL-17F genes. And what you can see are different tracks for different ChIP-seq experiments that we've done. So for example, these are 5 TFs that are very relevant for the Th17 lineage, as I explained before. This is P300 and CTCF as well. And what we see is that in many cases, we see them bind together around the same genomic region. And we asked how prevalent that is throughout the genome. Do we see it? What are the trends? What are the patterns of core binding for these transcription factors? And so what we've done is we projected all of these ChIP-seq tracks into one dimension, and then we can cluster and find those cases where all of these peaks, all of these binding sites fall in close proximity, and we call them putative cis-regulatory modules. And then we wanted to look at the patterns genome-wide, so we created this heat map here, where you have in the columns the different transcription factors, ChIP-seq data, including P300 all the way to the right here, and the different rows in each cluster here represent the different patterns that exist in the genome. For example, if we look at the first cluster here, this is a cluster that represents cases where all of the 5 transcription factors were binding together, so cases like this. And if we look, for example, at the bottom one here, we see a case where only MEF is binding in that region without the other transcription factors. So what you can note, for example, is that when they are binding together, all of them together, we can see a very strong binding of P300 associated with these genomic regions. We found it very helpful, so we developed a small little tool that takes ChIP-seq data, and it will create this kind of heat maps for you, giving you an indication of what is the pattern you have in your data, which genes, which transcription factors, which markers like to fall together with each other. And this was done together with Kieran May, who is now a PhD student at UCSF. And so what I want to focus on right now is just this section here. You see BATF and IRF4. They seem to co-localize a lot throughout the genome. And we wanted to explore this further. And so what we've done is we first looked for motifs. So what we've done is we looked for BATF, where BATF alone, what we found was the BATF motif, as you expect. When we look at places where IRF4 binds alone, we found the IRF4 motif, the canonical motifs for each of them. However, when you look at where they bind together, we actually find a composite motif that is otherwise not recovered. And what you see is that there are two versions. One is you have the BATF site together with the half site of the IRF4, either with no spacer or with a 4 base pair spacer. So they seem to have a different binding motif when they bind together. We checked if it's functional. I'm just showing you some of the results that we've done for this. We checked if this is functional by doing IRF4 ChIP-seq in BATF wild-type versus knockout, and reciprocally doing BATF ChIP-seq in IRF4 wild-type versus knockout. And here I highlight a few regions where they bind together in wild-type. However, when you knock out the other partner, you see that you abolish the binding site for the other TF, suggesting that they are functionally helping each other binding. And this is true genome-wide, although I'm just showing you here one case of that. In fact, this was published in the last month in different contexts of the immune system and was shown to be a very important interaction. Another small result that I want to show you that I thought was interesting is that if you just look at the relative position in these regulatory modules that have all of the 5 TFs together, what we found is that BATF and IRF4 are actually more central to these regulatory regions. They found in the middle compared to CMF or STAT3, where you see they are more peripheral or in the outside of these regulatory regions, where O gamma shows no preference. This may suggest that BATF and IRF4 actually actually establish these regulatory regions to begin with, and we have some data to support it that I'm not going to go into details here. What I want to do next is actually switch gears from the ChIP-seq data and go to the network inference. So we have a computational pipeline that is integrative in nature. What we want to do is, as we discussed before, we want to take the ChIP-seq data, the knockout data that we have for a few of the transcription factors that we know are important in the lineage. We want to complement it with the transcriptomic data that we collected, or we used the public resource. And what we're doing here is we say, OK, each of these sources of information give you an indication of a regulatory interaction between one transcription factor and all of its possible targets. And if we use a dedicated method to learn these regulatory associations from each data type alone, we can assign a confidence score for each of these regulatory interactions. And then we can rank for each transcription factors based on each of these data sets the regulatory interactions in order. And by doing this ranking, we can combine them based on the ranks to create confidence scores for each transcription factor and gene based on the combination of all data types together or based on any subtype of combinations. And what I want us to focus on here today is how well did we combine the data. Was it indeed mutually reinforcing, creating higher accuracy, or was it just reducing the performance of the method? And so when you do something like that, what you will get is a regulatory network. What we see here are the 5 transcription factors that we knew were important and we have knockout and ChIP for. And what you see here is, for example, the green edges represent activation, the red edges represent repression. So for example, you can see MEF is in general a repressor, which wasn't known before. STAT3 is in general an activator. The nodes are shaded in blue or orange to represent activation or repression respectively. These genes in the periphery are strong targets of only one of the transcription factors, whereas genes in the middle are strong targets of a few of the transcription factors. And now when we zoom into the middle of this regulatory network, we find all of the genes that we expected to find based on what I showed you before, the prior knowledge that we have, and they're indeed regulated by all of these transcription factors. However, we find many more genes that we thought are important. So before we go and start validating things, we decided to use this field is very active. A lot of information was gained by the time we created this network. So we curated the Th17 list of important genes, and we checked for the recovery of these genes by the network created from different combinations of data. So if you look at the blue, these are the results. Area under precision recall is just a measure of how well we recover these genes. If you look at blue, this is when we use one data type. This is when we use two data types. This is when we use all of the data together at the end here. And what I want you to take, the take-home message of this is, as we combine data, we do better and better in recovering the regulatory interactions. And so just another measure of performance. This is for the combination of all the data. This is something maybe you're more familiar with, GeneSnet enrichment analysis. And you can see that if we rank all of the genes based on their score in the network of combining all of the data, you see that we recovered all of the important, many of the relevant Th17 genes as high predicted targets. And indeed, we find many more that are not shown in the literature and we validated with knockdown, chip, or other methods. And what I want to focus on last is just connecting this network to GWAS data. So we wanted to see how well it connects to GWAS SNPs that are related to autoimmune diseases. We know the Th17 lineage is important for these diseases. So what we've done is GWAS data connects SNPs to disease. We just took it a step further and connected SNPs that are associated with genes that nearby within 100 kb. We associated the genes with the SNPs with the disease. So now we have, for each disease, we have a list of genes that are important for the disease. But they have no context. It's just a list of genes. And this is complicated disorders and conditions. And so if you do something like that, and we check for the recovery of these SNPs-associated disease genes, so in the left here, you see Alzheimer's, schizophrenia-related genes, and you see that the network doesn't enrich for any of these genes. However, when you look to the right here, you see that in Crohn's disease or in type 1 diabetes, you see a high enrichment of the network for these genes. We can look at the actual networks and see that in Crohn's disease, we have a very tight regulation of these genes here by the 5' transcription factor in Th17, and in type 2 diabetes, we see that it's not so important. So now we connected many of the genes in the GWAS study with a context. Maybe their effect is through their role in Th17, in the Th17 lineage. And with that, I would just like to summarize some of our findings. I showed you some things for IRF and BATF, but we validated many more transcription factors. We identified a plasticity modulator. And we also found that the linear specifier is really more of a modulator rather than a master regulator, which is supported by recent publications that show it for other linear specifiers of the TL per subsets. And with that, I would like to thank the people involved and to take questions if you have any.
No, please. We could start switching on. OK, perfect. So my question is the following. So for the GWAS analysis, this is very exciting that you're finding that indeed many of your top hits are in fact falling in sort of pathways. My question is, can you now exploit that to look beyond the significance threshold for additional genes that may have not made it there, but are still belong in the same category?
So I think that's a great idea, and that's where we're thinking about going in it. So one of the problems with GWAS studies is that you don't have the statistical power to really resolve many of the other genes that may be implicated. But if you have now the network, you can limit it to the areas where you think there should be something interesting and not test the whole genome, which there is a review by Andrea and Eric Schatt recently that suggests a similar concept. I think we should take it further and look at the actual regulatory regions instead of the genes that are important for these diseases, because now we can do it. We have the ChIP-seq. We know which enhancers are really important, so we can take these SNPs. Maybe they don't fall exactly in the right enhancer, but they will be in linkage disequilibrium with this enhancer, and we can really associate them with what they're actually doing, how they affect the...
And did you only use type SNPs, or did you actually use imputation to actually identify additional fine mapping? Because in order to do that, you would need imputed...
We didn't. So this is part of a large work. We didn't have... It's not this paper. This is just a small panel in this result showing that we can find these GWAS genes. But yeah, going forward and looking at it in more depth, this would be something to look at.
Great, thank you.
Thank you very much.
And let's thank Aviv again.